we are looking at the absolute limits of biology, specifically how plants manage to survive in the most extreme, unforgiving environments on Earth. I'm going to be guiding you through the essential, step-by-step physiological facts and the foundational theories you really need to master this topic.
And I'll be right here to play devil's advocate and challenge those theories. Because let's be real, textbook definitions are great, but proving them in the lab is a completely different ballgame. We're going to take these pristine concepts and put them to the test with real control groups and raw experimental data.
So let's just scrap the standard introduction and jump straight into the central challenge of this explainer. It's super easy to just say a plant adapts to a scorching desert or a flooded swamp, but how do they actually pull that off on a cellular level? And more importantly, how do we prove these survival mechanisms are real when we're in a controlled laboratory setting?
Okay, that is a totally fair challenge. But if we're going to prove anything, we first need to establish our foundational terms, right? We can't really run an experiment without knowing exactly what biological process we're even testing.
And that brings us to the core concept that's absolutely required for any board exam: osmoregulation. Basically, this is the physiological regulation of water and solutes to maintain homeostasis. It is the master balancing act. Literally, it's the difference between thriving and dying. Whether a plant is completely submerged in a swamp or baking in an arid desert, it has to regulate water loss and solute uptake if it wants to survive, grow, and carry out photosynthesis. All right, that's a perfectly neat textbook definition, but maintaining homeostasis looks wildly different depending on where a plant actually lives. Let's look at the physical hardware these plants use to pull that off.
These two microscopic cross-sections brilliantly illustrate that exact point. Take a hydrophyte, a water plant called Eichhornia crispus. If you look at its roots, the middle cortex is completely full of parenchyma and lacuna. Those are basically large intercellular air cavities. They provide crucial buoyancy and a rapid pathway for gas exchange, which makes sense because in that environment, water's everywhere, but oxygen is super scarce. But then you switch gears to a xerophyte, a desert-adapted plant like Phalangiopsis, and it's a striking contrast. Instead of open air pockets, you're looking at a huge parenchymatous cortex. It's essentially a giant cellular sponge specialized for one single purpose: hoarding and storing as much water as possible.
Okay, I'll admit the anatomy is fascinating, but anatomy is static, right? It takes generations to evolve. My question is, what happens when a perfectly happy, well-watered plant is suddenly hit by a totally unexpected drought? How does it actively respond in real time before it just completely wilts and dies? To answer that, we have to move from static anatomy into the world of dynamic biochemistry. We have to talk about chemical signaling.
The textbook answer to sudden water stress is abscisic acid, or ABA for short. Think of this as the premier plant stress hormone. When the soil starts to dry out, ABA is synthesized right there in the dehydrated roots. It acts like a biological 911 distress signal, traveling straight up the plant's transpiration stream to the leaves. Once it gets there, it forces the guard cells to close the stomata, which instantly puts a lock on water loss.
All right, so you've stated the theory. ABA from the roots closes the stomata in the leaves. Let's see how this holds up when we actually test it in the lab. How do we know for sure that ABA is truly the trigger here?
To prove this, researchers set up a really strict experiment. First, they have a control group, well-watered plants just living a completely stress-free life. Then, they have the experimental group. These plants are subjected to drought stress, but the researchers introduce an independent variable, applying a very specific concentration, 5 micromolar, of synthetic, exogenous ABA directly to the leaves. If your textbook theory holds up, this synthetic application should trigger some massive physiological changes. And wow, the lab data reveals things the basic textbook definition totally misses. When that synthetic ABA was applied, leaf conductance literally plummeted by 50% in detached shoots. The stomata just slammed shut. But interestingly, the ABA also triggered the accumulation of dehydrant proteins. These actually physically protect the cellular structures from dehydrating. But here is the coolest part. As the drought sets in, the xylem sap actually becomes more alkaline, shifting from a pH of 6.3 up to 7.2. Why does that matter? Because that specific pH shift prevents the ABA from being absorbed by random mesophyll cells. It forces the hormone directly to the guard cells where it's needed most. The chemistry perfectly dictates the survival response.
Okay, so we've conquered drought, but let's flip the script entirely. What happens if the environment goes to the complete opposite extreme? What if the roots aren't dry at all, but are completely buried in dense, flooded, anaerobic mud where oxygen diffuses about 10,000 times slower than it does in the air? Well, in Matt's scenario, saving water isn't the problem, saving breath is.
Without specialized adaptations, the plant will literally suffocate.
To survive being completely flooded, hydrophytes rely on parenchyma, which we touched on earlier. If you look at a microscopic view of a Ludwigia repens stem, you can clearly see the uniseriate parenchyma. It's essentially a single layer of thin-walled cells creating wide-open gaps. They act exactly like a built-in snorkel. Because the roots are trapped in a totally oxygen-free zone, this internal highway establishes a crucial oxygen gradient, letting oxygen travel from the leaves above the water all the way down to the submerged root tips. A snorkel is a great analogy, but a snorkel is completely useless if it has holes in it. If the oxygen leaks out of the root into the muddy soil before it ever reaches the growing root apex, the plant dies. So let's look at the data on how they actually prevent that leak. The data on radial oxygen loss, or ROL, really tells the whole story here. True wetland plants, like Oryza sativa, which is rice, have high oxygen loss right at the root tip, around 175 nanomoles. But look higher up the root, almost zero loss. They've developed a tight anatomical barrier in those basal zones. This barrier perfectly seals the root, funneling all that precious oxygen strictly to the apex so it can keep pushing into the anaerobic soil. Now contrast that with triticum, your standard non-wetland wheat. It has a completely weak barrier. It leaks massive amounts of oxygen high up the root. So if you flood standard wheat, it bleeds its oxygen into the mud and drowns, which is exactly why flooded fields absolutely destroy global crop yields.
Okay, textbook expert, I'm taking us back to the desert for the ultimate biological paradox. To survive the blistering heat, a plant absolutely must close its stomata so it doesn't lose water. But to perform photosynthesis and actually, live, it must open its stomata to absorb carbon dioxide. So how on earth do you solve a problem where the very act of breathing causes fatal dehydration?
It's brilliant, actually. You solve it by separating your physiological processes, not by space, but by time.
So the crucial concept here is the CAM photosynthesis pathway, crassulacean acid metabolism. It's an incredibly elegant, two-step time-shifting process. Step 1. The dark phase. At night, when temperatures drop and it's nice and cool, the plant opens its stomata. Carbon dioxide enters, gets fixed by an enzyme called PEP carboxylase, and is safely stored away in vacuoles as malic acid or malate. Step 2. The light phase. When the hot, scorching sun rises, the stomata lock tightly shut to prevent any water loss. The plant then takes all that stored malate, breaks it down or decarboxylates it to release the CO2 internally and runs the Calvin cycle. They literally just hold their breath all day long. And we have the visual proof for this too. When you take a stomatal peel, which is basically a microscopic surface imprint, and compare a water-loving plant like Ludwigia with a CAM desert plant like Crassula ovata, the difference is night and day. The hydrophyte is absolutely loaded with stomata because it can afford to lose water. The CAM plant, on the other hand, heavily regulates its stomatal density. It's perfectly engineered to operate on that highly specialized nocturnal schedule. Beautiful theory, beautifully explained. But I'm going to dial up the pressure one last time. What happens when you combine the need to conserve water, the constant threat of intense heat, and toxic, cellularly destructive levels of salinity? I'm talking about the ultimate boss level. Halophytes, living in estuaries. How do mangroves survive pure seawater?
Mangroves, like the species Avicennia marina, are the ultimate survivors. They use this amazing multi-tiered defense system. First, they perform non-metabolic ultrafiltration. This acts like a physical sieve right at the root level to aggressively block salt from entering. Second, they carefully manage the movement of whatever liquid water and ions do slip through via the transpiration stream. And finally, they feature specialized excreting salt glands and these blister-like vesiculated trichomes on their leaves to just physically push excess salt right out of their bodies.
Okay, ultrafiltration and salt glands. Sounds good on paper, but how much salt can they actually handle before the whole system just breaks down? Let's take them into the lab.
Here's exactly how we test those limits. We place these mangroves into varying substrates, 0%, 50%, and 100% pure seawater. Then, we strictly measure 3 dependent variables: how much salt gets into the roots, how much actually makes it all the way up to the leaves, and the diurnal rhythm, the daily timing of their salt excretion. Let's look at the numbers. The data here is just spectacular. As the seawater concentration goes up, the salt inside the roots increases on a perfectly linear scale. Even at 100% seawater, which is a massive 450 millimolar of sodium, the roots haven't maxed out. They just keep taking it in. But the leaf salt response?
That tells a totally different story. It hits a hard plateau at about 1.4 millimoles per gram of dry weight. How is that possible? Because the excretion rate spikes to match the intake. And surprisingly, they excrete minimally during the day and maximally at night. This proves that the ultimate control of salt content doesn't actually happen down at the root. It happens at the foliar, or leaf level. The leaves act as the final, absolute gatekeepers against toxicity.
From osmoregulation to the CAM pathway all the way to the incredible salt glands of mangroves, it's just so clear that nature has already engineered brilliant solutions to the planet's absolute harshest extremes.
Exactly. And the lab data proves that these mechanisms are highly responsive, chemically driven, and incredibly effective, which really leaves us with one final, massive question to think about. With rising global temperatures and shrinking freshwater supplies, could we actually bioengineer these very pathways, things like radial oxygen barriers, ABA sensitivity, or foliar salt excretion, to create the drought-proof, salt-tolerant crops of our future? It really is the ultimate test of translating plant physiology from theory
into practice.
Thanks so much for joining us on this explainer.